Thanks everyone for coming. It's my real pleasure to introduce tonight's faculty lecturer, Pat Griffin, who's professor and chair of the Department of Molecular Therapeutics. Pat received his scientific training in two of the most prominent labs in the field of large-scale or systems biology, a PhD with Don Hunt at the University of Virginia and a postdoc with Lee Hood at Caltech. If this educational course sounds vaguely familiar to some of you, it's because it mirrors that of our colleague John Yates, with whom Pat has forged a long-standing and productive collaboration and friendship that began in grad school and ensues to this day. I can honestly only imagine how exciting it must have been to have been part of Hunt and Hood's labs in the late 1980s and early 1990s when mass spectrometry was just beginning to show signs of becoming what we all now recognize to be the transformative modern technology for protein science and proteomics. In the Hunt and Hood labs, Pat contributed substantially to the advancement of both experimental and computational approaches for mass spectrometry-based proteomics, and after completing his postdoctoral studies, was quickly snatched up by Merck Research Laboratories, where he rapidly rose through the ranks to become a senior director of basic chemistry and molecular profiling proteomics. Pat's fingerprints can be found on some of the most seminal discoveries to have emerged from Merck in the past 2 decades, perhaps most notably in his collaborative studies with Don Nicholson and Nancy Thornberry and others to discover the Cas-based enzymes and their roles in apoptosis, and dipeptidylpeptidase 4 as a new therapeutic target for type 2 diabetes. It was during a visit to Merck that I first met Pat. We had a really stimulating conversation that traversed a dizzying array of scientific subjects ranging from next-generation proteomic platforms to the chemical biology of enzyme inhibitors to the challenges that face drug development in both academia and industry. I came away from that meeting with one simple impression, that this guy was far too creative and dynamic to remain at Merck much longer. That premonition proved correct because shortly after my visit, Pat soon departed Merck to become CSO of XR Corporation, a biotechnology company focused on using hydrogen deuterium exchange mass spectrometry for drug discovery. In 2005, I believe, is that correct? Okay, 4. Your CV is opaque in this regard, but somewhere between 2004 and 2005, we were extremely fortunate to recruit Pat to TSRI, Florida, to become the founding chair of the Department of Molecular Therapeutics. To say that Pat's academic career has gotten off to a fast start would be a gross understatement. I think it's unsurprising to anyone who already knew Pat. He immediately built a vibrant research program at Scripps, Florida that leveraged his vast knowledge of basic and translational sciences for compelling problem selection and his penchant for developing innovative methods for the discovery, mechanistic characterization, and biological application of enabling chemical probes. Pat's interests are certainly diverse, but remain firmly grounded at the interface of chemical biology and mammalian disease. He has made landmark contributions to the development of probes and biophysical methods to study nuclear receptors and their regulation by signal transduction pathways with relevance to metabolic disease, cardiovascular disorders, and infectious disease. A subset of these findings have formed the basis for a new biotechnology company called Ember Therapeutics, which Pat co-founded with Bruce Spiegelman to create next-generation drugs to treat metabolic disorders. Pat has also played a critical role in building TSRI's Small Molecule Screening Center in collaboration with Hugh Rosen, which is now widely recognized as the best in academia. And more generally, Pat has served as a pivotal figure in putting Scripps, Florida on the map in terms of both his own lab's research accomplishments and as a chair committed to building a top-notch research campus through recruiting and supporting the next generation of scientific leaders. Pat, it's a real pleasure that you could join us today, and we very much look forward to your lecture entitled Mechanism of Action of Novel Insulin Sensitizers.
Thanks, Ben. Hopefully I can live up to that introduction there. Yes, it's been a lot of fun in Scripps, Florida. It's been a journey, to say the least. I'm actually there now almost 9 years. It's hard to believe. It was only yesterday when I showed up. On the slide here, I did throw in a couple of other tidbits because I'm going to not only talk about insulin sensitizers, I'm going to touch on our work on the ROR receptors in terms of their effects in autoimmune and also in potentially in anti-obesity. Half of my lab focuses on this HDX platform, which we have over 50 collaborations worldwide. I'm not going to cover this in this talk other than give a little bit of intro into it because it applied, I'll show some of the data on how we applied it in the PPAR gamma program. So this slide summarizes really what our lab's all about. This is just a cartoon of a nuclear receptor. associated with DNA. And the simple models of the past of how nuclear receptors were activated just by ligand binding, some sort of conformational change in interaction with co-activators or co-repressors has just basically been exploded upon to now where there's multiple post-translational modifications that control these interactions. These interactions can be quite diverse. They can interact with different regions of the receptor. So basically, we view this as combinatorial control for nuclear receptor function. These just give you a list of some of the types of functional interactions that one could focus on. So really, the goal of the lab is to develop functional selective modulators of NRs. So basically, if this phosphorylation event has a specific impact on function of a receptor, can we go after targeting that selectively and looking at what the biology is downstream? And then, as I mentioned, we've developed this HDX platform, which, again, won't be the subject of the talk. What I'll focus on in the first example is really ligand influencing on PTM status of a nuclear receptor complex, and then how that post-translational modification affects co-regulator interaction. So just brief background, again, with a diverse audience, just a little bit of information here. The nuclear receptor family has this canonical domain structure. All with the exception of 2 of the 48 members have DNA binding domains. The remaining 48 have ligand binding domains. And there is communication across all these domains. And the receptors can function as monomers, homodimers, or heterodimers. And the 2 examples I'm going to talk about on the RRs, particularly RR gamma, is a monomeric nuclear receptor, and then PPARs are obligated heterodimers with RXR. What I tell my lab, postdocs entering the lab, while we focus currently on 3 receptors in particular, we do have a lot of work on vitamin D receptor, progesterone receptor, et cetera. Given the diversity of the family, the way this alignment is that the highly conserved region is the DNA binding domain, but all the other aspects, the N-terminal domains and the ligand binding domains, have a high degree of diversity. So what I tell them is we're never going to run out of things to do, because in almost all cases, the function or role of this AB domain is completely unknown. So just a very high-level crude model with a million exceptions as to the rules here, but in general, if you just view the nuclear receptor complex associated with a response element in the promoter region of a target gene, it's associated with a co-repressor complex which has histone deacetylase activity associated with it, and it's basically not allowing transcriptional machinery access to this promoter. If we add its natural ligand, if it's an agonist, it would dissociate the repressor, bring in the co-activators with histone acetyltransferase activity, unravel this region, and allow the basal transcription machinery to come on, and you get transcription on. So that's the, again, really high level of how nuclear receptors go. So since I can only focus on a few aspects of what the lab is doing, I just have a few highlights in the last few years. In the case of the PPAR area, we basically had uncovered a mechanism on how the current anti-diabetic drugs actually impact PPAR gamma, and we've been utilizing that information to develop a next generation series of molecules. There were 3 key papers here. The structure paper in 2007 is extremely highly cited, and it basically provided the insight for, or interest for Bruce Spiegelman to focus on this particular region of the receptor, and that led to back-to-back or 1 year subsequent papers in Nature, one where we described the mechanism and then one where we described the development of the novel modulator. I will go through this in some detail and then get into the next generation compounds that we're working on. In the RRs, we basically were the lab leading the sort of not de-orphanizing of this, but finding the chemical modulators. There's a series of papers published and then developing basically isoform selective potent molecules, and showing that at least one of these compounds was efficacious in an EAE model for MS, and that was published in Nature in 2011. And then I'm going to show you the data here on CIA. I'm going to skip this part of the story, but then go into this latest observation, which was actually unexpected, where one of these compounds shows robust anti-obesity effects in DIO mice. LRH1 is another receptor of interest to us, but because of time, I'm not going to cover this at all. But basically, we found molecules that are potent at removing the receptor from chromatin and basically are functional inverse agonists without directly binding to the receptor. We've shown that these have been efficacious in various cancer cell lines, and in collaboration with David Moore, showed that DLPC is in fact a bona fide agonist of the receptor and shows improvements in hepatic steatosis, and that was also published in Nature. So the presentation is as such. I'm going to talk about PPAR gamma. This is a receptor with a very long history, but I'm going to be focusing on a specific post-translational modification. And then what we now coin, the selective modulators have been the terminology used for nuclear receptor compounds that do things that we're not quite sure how they do it. Functional selective, we'll get into how can we functionally modify specific regions. And then in the RR case, I'll go through the CIA model and this anti-obesity effect. And as I mentioned, due to lack of time, I can't cover the LRH1 story, but also our extensive work on HDX, on GPCRs, kinases, and nuclear receptors. We'll skip that. So a little bit of background on metabolic disorders and what we're actually trying to do with these particular compounds in the PPAR areas. We're looking to improve peripheral insulin sensitization. It's a multi-organ disease where you have effects by deficiency in insulin signaling, resistance to glucose uptake due to peripheral insulin resistance in muscle, increased hepatic glucose production, again being unresponsive to insulin signaling, and elevated free fatty acids. So the reason for pointing this out is that in the studies I'm going to show you, we have to measure effects of compounds in multiple tissues. There's a clear causal link between obesity and type 2 diabetes, and this is BMI among adults diagnosed with type 2. You see that there's only a very small number in which are considered normal BMIs, whereas the majority of type 2 diabetics have BMIs that put them in the overweight or obese category. And the reason for pointing this out is that we're going to use diet-induced obese mouse models where we put these animals basically on an Atkins diet of 60% fat to induce insulin resistance. So the question is, and we all know there's an obesity epidemic, and I'm not going to touch on exercise here, but clearly there's a link between exercise or activity and BMI. So this is often referred to as the evolution of inactivity. So we're typically now here, we're no longer hunting and gathering, and we're not building things. We're sitting on a computer all day, and our steps per day have reduced dramatically over the years. But so why do we need effective therapies for type 2 diabetes? And the complications are listed here. Some of these are well known and real obvious, and some of those are severe and irreversible, obviously. And so even with diet and exercise and the current therapies, these comorbidities have increased over the years. There are many approaches to treating type 2 diabetes or frank diabetes in the case of insulin when there's a complete loss of beta cell mass. On the insulin resistance side, there's really just the TZDs. Metformin can be classified as an insulin sensitizer, but it's really blocking hepatic glucose production. You have the incretins and the DPP4 inhibitors for impacting beta cell dysfunction. But the bottom line is, in the absence of a disease-modifying therapy, you ultimately end up using insulin. And that's pointed out here. There's a need to intervene early. The earlier you intervene, if you can intervene between the prediabetes and the frank type 2 diabetes, you can actually have a disease-modifying effect where this will not progress further. And TZDs have shown robust clinical efficacy and have shown significant ability to block progression of disease. And then I'll get into the reason why, even though this is fantastic, there's a lot of problems associated with TZDs. So that's shown here. The two compounds that have were registered and had billion-dollar sales. Many of you might be familiar with Avandia. You might have seen ads where the lawyer said, if you've taken this, come see me, you'll make a lot of money, because you are at risk for many diseases, or particularly if you have had a heart attack. These are very efficient at controlling fasting plasma glucose and reducing glycated hemoglobin. But there's a weight gain, a dose-dependent weight gain associated with an edema or a plasma volume expansion, which is believed to be the leading cause for the cardiovascular issues, leading to increased work on the heart. And because of this phenomenon, there's been significant black box warnings. In particular for Avandia now, it's been withdrawn in many countries. So while it being very efficient in the therapeutic side, it has significant issues. Now, pioglitazone, if you look at these structures, they're extremely similar, but because this is slightly potent and has slightly less poorer pharmacokinetics, the actual number of issues with the cardiovascular events has been lower, but still, because of the class, it gets the black box warning. But I think the clear thing is that it's not a class effect, it's really a compound-specific effect, and in fact, PIO has a unique issue with bladder cancer, and this is believed to be unique to PIO in that its poor solubility leads to mechanical or precipitation in the bladder and mechanical issues, causing the increase in bladder cancer. Now, there are multiple isoforms of PPAR, just pointing this out because there could be distinct tissue distribution, but the roles of the two major isoforms we're going to talk about today, alpha and gamma, where alpha is really involved in fatty acid oxidation and triglyceride modulation. PPAR gamma is really involved in adipogenesis and insulin sensitization. But a critical thing is this bone function. In fact, I mentioned it on the previous one, that there's issues. It says, so the negative effect means it actually has, impacts bone density, and I'll get into this briefly later on. So people have, there's probably hundreds of papers in the literature of how TZDs impact insulin resistance. One thing that's clear, if you put mice on a high-fat diet, you get suppression of two adipokines, adiponectin and adipsin. These are molecules that are important in enhancing peripheral insulin sensitivity. And these are bona fide PPAR gamma target genes, so if you stimulate with agonists, you're going to increase an expression of these molecules. This is actually human data here showing patients on a TZD do have a statistically significant increase in adiponectin. However, there's hundreds of PPAR gamma target genes, so it's really hard to dissect any one specific effect from our target gene on what could be causing or driving the clinical efficacy. So in addition to that, there's some interesting questions. So partial loss of function mutations of PPAR gamma in humans lead to lean patients that have severe insulin resistance. So you would look at this and you would say, okay, if you activate PPAR gamma, you might overcome this issue with severe insulin resistance. However, the PPAR gamma agonists, they improve insulin resistance and diabetes in patients that are typically obese and have no defect in the receptor. So about 99% of the patients in which TZDs are used effectively, there's no mutation in PPAR gamma, and if you actually look at fat biopsies, the PPAR gamma target genes are fully on. So there's a disconnect between that. So you're not using the agonist to overcome a mutation, you're actually super activating the receptor that's already normal. More confounding is that if you develop weak agonists or partial agonists of the target, they have the same or equivalent anti-diabetic effect. So, why is this the case? So an example of such compounds, Merck had spent a lot of time on this, developing what they called SPPARMs for selective PPAR gamma modulators. And we have shown in others that the partial agonists have the SPPARM phenotype, whereas they improve glucose uptake, no impact on weight gain, plasma volume, et cetera. But the caveat is the side effects here vary from model to model, so DIO to DBDB to OBOB, as well as the duration of administration. And none of the papers focused on bone density because that was not an issue that had been presented yet in the clinic. So the questions about SPPARMs, what are the mechanisms of action for PPAR gamma modulators versus the SPPARMs? Are they different? And is it different than that of adipogenesis for the full agonist? And then what are the molecular determinants that differentiate the downstream effects? And what what got us intrigued in this from day one was the possibility, can you actually dissect the insulin sensitization from the pro-adipogenic fluid expansion, et cetera? So with the ultimate goal shown here, we know that TZDs are robust insulin sensitizers, the most robust insulin sensitizers available to the clinician. They can actually prolong the development of diabetes, but the side effect profile has really hampered the use of these molecules. So, as I mentioned, we got involved with Bruce Spiegelman, who had been focused. They had a postdoc in the lab that was showing a particular phosphorylation of the receptor seemed to correlate very well with repression of a subset of PPAR gamma target genes. Remember, I said there's hundreds. This subset is about 13 genes, and these 13 genes are the most downregulated in the obese state. So there seemed to be a connection between this phosphorylation and those genes. We worked out the mechanism that actually, if you, in the obese state, you have increase in pro-inflammatory cytokines that drives the processing or cleavage of the cofactor for CDK5. P35 cannot translocate, P25 can. So the kinase complex can translocate and phosphorylate the receptor. Then the next interesting thing we realize is that if you treat with PPAR gamma ligands, you can actually block this phosphorylation event and de-repress these genes with the model shown here. So when the receptor is phosphorylated, there's some cofactor, yet I'll show you in a few slides what we think it is, associated with the complex. It's repressing a subset of PPAR gamma target genes. If you add either a TZD or a partial agonist, you interfere with that phosphorylation. You dissociate that cofactor, and you then activate PPAR gamma target genes. So, however, though, you're still recruiting in excess or extensive amount of co-activators, so you're not only turning on this subset of genes, you're turning on many, many PPAR gamma target genes. And the data supporting this is shown here. These were some of the tool compounds we used, rosiglitazone or Avandia, the Merck partial agonist MRL24. This is a transactivation assay. You can see there's a clear difference in the degree in which Rosi can transactivate a reporter versus MRL24. But if you do dose diabetic mice and do the best you can do to normalize for exposure, these are equipotent in binding the receptor, and they have very similar pharmacokinetics. We get equivalent anti-diabetic effect and a GTT between those two molecules. But what was interesting when we looked in fat pads and looked at the ratio of phosphorylated PPAR gamma at serine 273 to total PPAR, there was essentially the same level. So the correlation between this efficacy and the blocking of the phosphorylation was much better than the activation. We also looked at human patients, biopsies from patients taking TZDs or diagnosed with type 2 diabetes on TZD treatment. And what you're seeing here is the response ratio to the drug plotted against the ratio of phosphorylation to non-phosphorylation. This was done in a blinded fashion. There were non-responders to the drug, and what you get here is a very nice correlation. So this was very encouraging that what we were seeing in mice actually has some correlation to the human condition. The other thing we looked at in a cellular model where we can knock out, this is a MEF cell line from PPAR gamma null mice, and we can put back in the receptor where we knock out the phosphorylation site at serine 273, and then look at what genes are differentially expressed. And what you see is there's a subset of genes that their expression when it's unable to be phosphorylated are increased, and we classify this as the serine 273 phosphorylation gene set. The classical agonist genes were unaffected by the phosphorylation site. So there's genes that are modulated by the phosphorylation. And then so we asked the question, is it possible to develop a compound that would block serine 273 phosphorylation and not activate the gene set? So again, the reason for this is that the genes that were corrected by the phosphorylation are the known factors involved in insulin, peripheral insulin sensitization, whereas the classic agonist genes are involved in adipogenesis. So again, Bruce and I worked on this, and our structural expertise in this area is extensive from 2007 on. And basically what we came up with with looking at a lot of compounds. So if you have a compound that binds just in the pocket of PPAR gamma, they're all partial agonists. If you have a compound that binds in the pocket and reaches over to what's called AF2 or helix 12, they're all full agonists. If you have compounds that bind more towards this beta sheet or omega pocket where the phosphorylation site is on serine 273, they block very well. So, however, if you have a compound that can reach over from this site and actually interfere with helix 12 and antagonize it, you have a compound that we're looking for. So a non-agonist, but agonist on the phosphorylation gene set. So to find these molecules, what we decided to do was fairly simple. We took a collection of all known potent partial agonists of PPAR, ran them in a binding assay, made modifications to these to increase the size or decrease them. And just as an example, the compound I'm going to talk a lot about, 1664, has essentially the same binding potency, the PPAR gamma. But when you put it in a transactivation assay, it's essentially dead. At 1 micromolar, there's no activity, whereas ROSI is significant. If you put it in full-length receptor context, it's flat as a board. This is the partial agonist MRL24. If you then look at its ability to block phosphorylation, you see a nice dose response. And then again, similar to the previous data I showed you with MRL24 versus ROSI, our antagonist CDK5 blocker shows equal efficacy to rosiglitazone when you normalize for exposure. So we were excited about that, but of course, you had to do further testing in the bona fide model for adipogenesis, differentiated 3T3L1s. 1664 looked no different than DMSO. You could ask the question, well, maybe this was not cell penetrant, and that's why you're getting these effects, but we can do a competition assay with ROSI, and it dramatically right shifts ROSI's EC50, so we know that's not the problem. TZDs are known to be anti-inflammatory. We show that 1664 has the same ability to block IL-6 production in a macrophage cell line stimulated with LPS, so we're maintaining the anti-inflammatory effect. This is in vivo phosphorylation data from 1664. Again, no difference from Rosie, and I showed in the previous slide similar efficacy. And again, a dramatic difference in transactivation, so the agonism is not correlating at all. And this is one of the side effects you can measure readily in mice. Rosie increases their body weights, and 1664 looked like a vehicle. And then the traditional oil red staining, we're not driving lipid accumulation in L1 cells. So the question, one of the issues with TZDs is negative on bone. This is a immortalized osteoblast cell line, and we're showing that rosiglitazone has a detrimental effect on 3 of the genes important for bone maintenance here, and 1664 looks no different than DMSO. And a marker for adipogenesis in bone is the increase in PPAR gamma, and we're neutral on that. So we're a non-agonist in the bone. 64 does not impact mineralization as well. So that's expression, but measuring alkaline phosphatase activity in these bone cell lines, the partial agonist MRL24 represses about 50%, whereas the full agonist Rosie is 90% repression, and 1664 is actually neutral on this. And again, we can do additional looking at pro-osteoblast signaling, et cetera. So these antagonists that block CDK5 phosphorylation are neutral on bone. So if we had to summarize 64, SPPARMs, so TZDs were the first generation, SPPARMs were the second generation. They're both pro-adipogenic, although they do block the serine 273 phosphorylation, and we're suggesting that that's why they're actually or improve insulin sensitization. They block, they're an NF-kappa B antagonist through this sumylation, which I didn't discuss, but 64 is. And then 64 is the only bone sparing. So that's why we're using this term as a functional selective PPAR gamma modulator, just for the sake of coming up with a new acronym. So then the main question is, well, what does the phosphorylation event do to the complex? So using this null MEF, we transduce with these different receptors. We can pull them down, run gels, cut out the bands, ID proteins here, and THRAP3 was identified as being present when TNF was stimulated in these cells, and it was absent in the mutation. We could do pull downs, co-IPs, to show there's an interaction only in the presence of TNF, meaning that when the receptor is phosphorylated, you have an association with THRAP3. And then we show that the compounds can block this and reduce THRAP3, as well as block the phosphorylation, and THRAP3 concentration is unchanged. So THRAP3 is a factor that binds the PPAR gamma when serine 273 is phosphorylated, and we're doing functional studies ongoing. But just to tell you, THRAP3 is a thyroid receptor active interaction protein, so it's a known interactive for nuclear receptors. So at least in that context, it makes sense. So the model here would be that when the receptor is phosphorylated, THRAP3 is associated. You put in a compound like 64 that only blocks the phosphorylation. We dissociate THRAP3, but we're not driving interaction with co-activators, so we only increase the expression of the target genes that are de-repressed or repressed by the phosphorylation event. So we're not getting this hundreds of genes turned on, we're correcting 17 genes. And to our knowledge, this is the first example of using a ligand binding pocket of a nuclear receptor to allosterically block a PTM as its major means of activation. And when we look at this from a therapeutic index standpoint, what we're saying is that to have efficacy here, you need to block the phosphorylation event. The higher degree of the agonism, the worse the therapeutic index. So as you go from a full to a moderate to a partial to a non-agonist or an antagonist, you should improve the therapeutic index. So how did we get to this compound? So as I mentioned, we started with potent partial agonists, in this case, this GSK compound, that's a super potent partial agonist, and we came up with 1664. those of you that look at chemical structures all the time, you realize that these are very subtle changes between these two. We've added substituent here at the 5 position, and instead of the racemic ethyl group here, we went to a S-methyl version of this compound. And when you look at the transactivation activities of these, they're dramatically different. So rosiglitazone is here. This GSK compound is super potent in terms of EC50. It's not a full agonist, but it's a moderate to partial agonist of the receptor. These subtle changes here cause it to basically be inactive. This little tick up here is an artifact of the GAL4 system, because every time we run these in full length, we see no agonism at all. But it's not too surprising given these are chimeric receptors. So we wanted to understand the molecular basis for this functional selectivity. So we did the co-crystal structures of all 3 compounds, including the R enantiomer 63. And when you look at them, for the most part, they look identical, but there are this subtle difference in distance of this part of the molecule. So the co-crystal structures, at least from the carbon backbone, looked identical, but there was a difference in the positioning. And then again, when you look at the transactivation for the R enantiomer, it's almost a full agonist. So this single change between these 2 molecules causes this dramatic difference. So the question is, what's going on? And we can show this not only in a reporter assay, in terms of AP2 expression in L1 cells, 63 is a full agonist and 64 is a non. If we do the standard stuff one does in the nuclear receptor field, look at peptide interactions, 64 is not recruiting a peptide that is associated with the NCOR protein, whereas, or dissociating it, sorry, whereas rosiglitazone does a robust dissociation, and so does 63. And you can look at co-activators, and they're different. So the question is, can we use HDX to obtain insight into what's going on here? So for that, I got to give a brief background. So hydrogen deuterium exchange is basically taking a protein and heavy labeling it with deuterium. And in this cartoon, I'm showing you the blue circles here relate to the amide hydrogens between the peptide bonds. And that's what we're actually measuring in this experiment. You expose it to heavy water, regions of fluctuation or regions that are stable will show differential incorporation of deuterium. We built an extensive platform in Florida. We have 3 systems completely dedicated to this. And as I mentioned, we have about 40 collaborations here cranking out quite a bit of data. And the way the experiment works is you expose your protein over time to deuterium. You have to then freeze that reaction, and so this you can do under whatever conditions you want. You want to lock in the deuterium, and then you want to denature and fragment the protein and preserve deuterium. So to do that, we have to do this at low pH. It's an acid-base catalyzed reaction with the minimum at 2.7. So we do that, and then we put it over a pepsin column, fragment it, and then let the mass spectrometer do the rest. So here's an example of a protein peptide without deuterium, and as you incorporate deuterium, the centroid of the isotopic envelope shifts up in mass. We use software to calculate that centroid and the percent D. In this case, here's a peptide from a receptor or protein without ligand, and then in the presence of ligand, you see this change in the rate and the difference in area under the curve. We can plot it many different ways, overlay it on a heat map, overlay it on the crystal structure. And just what does this mean? So if we look at the vitamin D receptor interacting with its natural ligand, every single hydrogen bond can be accounted for significant protection to exchange when you take the APO receptor and add it. But then there's regions that don't immediately, it's not immediately obvious as to what's going on. Turns out these are the dimer interface between the homodimer, or in this case, VDR and RXR. So you get information of direct binding as well as allosteric interactions. The precision of this system is extremely high. If we, looking at over an 8-month period, just one system, 30-second on exchange point, 127 replicates, we get about 4,000 out of 4,100 measurements within 10% of the mean. So you can rely on this data such to the point where you can cluster a series of ligands based on whether or not the change between the APO and the receptor were statistically significant. So the blue color is with a p-value less than 0.05, and then we can cluster these fingerprints. Just so you get an appreciation, this was 3,654 injections in the mass spec, or 300,000% D values represented in this table. And we're perhaps the only lab capable of doing this magnitude of HDX. So what does this mean, though, in terms of being able to use this for understanding mechanism or using it to look at compounds on how they function? In collaboration with Eli Lilly, we were using HDX to help their vitamin D receptor program. There they want to have compounds that have very potent activity on a bone hormone known BGLAP. So all these compounds are active on BGLAP, which is a bona fide vitamin D receptor target gene. And then there's TRPV6, which is a PPAR VDR target gene that is calcium channel and related to the dose limited toxicity. So we simply ask the question, if we look at one region of VDR, look at its HDX characteristic, can we make sense into whether or not they would be a good V-derm or a bad V-derm? And in fact, that's the case. There's multiple peptides in the receptor that do this correlation, and now Lilly uses this as their primary assay. They send us 20 compounds a week, we return the data, and they decide which compounds go into the secondary assays. So in the PPAR story, back to this, you can't read any of this, but the bottom line is these antagonists showed a unique pattern. When you overlay it, it's in the business end of activation of the receptor. So what we decided to do is mutate in that region. So if you make mutations, you can cause ROSI to become a non-agonist. The partial agonist MRL24 is a non-agonist now. We show in binding assay that we didn't interfere with the ability of these compounds to bind. And this is the region here, the amino acid that we're mutating, which is in the co-receptor interface. And then we've subsequently shown that this residue actually influences the interaction between the co-receptor. So the other thing we're able to do with the HDX data is show that, again, another key business end of the nuclear receptor is helix 12. And we show that there's a nice correlation between activation of helix or stabilizing helix 12 and agonism. So we have several models now to filter this out. Now, as we were going along doing SAR, looking at more potency, we were making more potent compounds, and then looking in reporter assays, we found for the first time an interesting phenomenon that we were actually getting a dip in the response in the reporter assay. So we were thinking perhaps this was inverse agonism. We proven that in the differentiating L1 cell assay, where it actually represses AP2. It can block differentiation of adipocytes. And this is actually bad. You would think maybe this is good, but you actually need adipocyte expression to have adipokines, which are the peripheral insulin sensitizers. So if you put this compound, which is an inverse agonist, in mice, you actually make them sicker. So their fasting plasma insulin is worse than that of controls, where you see ROSI having the expected correction. So the question is, can we use HDX to filter and avoid developing inverse agonists? And that in fact is the case. What we see here is that the percent stabilization of helix 12 correlates with agonism, where 1664 is neutral, there's no stabilization, it's a non-agonist. If you start increasing agonism, protection of helix 12, you have inverse agonists. And then we actually have two different ways to get to inverse agonists. If you actually destabilize helix 12 significantly, you also have inverse agonism. So a combination of HDX and a lanthoscreen-based assay, I'm not going to go into detail here, but it's clear that looking at certain nuclear receptor co-interactors, you can clearly distinguish non-agonists from inverse agonists. So if we summarize 1664 as a functional SPARM. This compound has a similar KD to PIO. It doesn't drive AF2 agonism. It's inactive in reporter assays, doesn't alter agonist genes in cell culture and in vivo, and it blocks serine 273P, and it's anti-diabetic. So it's an antagonist of endogenous ligand, but an agonist of the serine 273 phospho gene set that's repressed. So the compound disrupts the receptor, and as I mentioned, disrupts the CoA interaction with RXR. So we have a lot of SAR around this, but questions remain. Will they have the same anti-inflammatory properties, which clinicians think are critical for the glitazone's efficacy? TZD's brown, white fat, or beige cells, partial and non-agonists, as published to date, have not been able to do this, and will this limit efficacy? But the real question for us, will the FDA tolerate a new PPAR gamma molecule even if you can prove a distinct mechanism, which I think we proved? So luckily to us, this had been dormant since 2009, reemerged as a series of announcements by Roche, where they have a large phase 3 trial with 19,000 patients with alglitazar, which is a full agonist on PPAR gamma and an agonist on PPAR alpha. And they have a glycemic control type 2 phase 2 trial as well, with the data expected. That's when this was presented, the design. The data is anticipated in early 2014. I'm not going to go into this, but we see they saw a dose-dependent increase in edema associated with this compound, and that is in fact one of the rate-limiting toxicities. So if they get approval for this, because they're showing a benefit in glycemic control, but they're also showing a reduction in cardiovascular issues in patients that have already had heart attacks. And that's mainly due to the fact that this does a robust job at improving lipid profiles. So they announced this, that they expect in 2014 they can launch. So that got us excited. This is our compound progression plan. I'm not going to go into any detail, but just to point out that my lab does all these boxes here. So there's a lot of stuff that has to go into the next few slides I'm going to show you. Luckily, we have a DMPK core that can do both the mouse PK, full rat, and some of the other important aspects. So I'm going to go through the current generation of compounds and try to do this quickly so I don't run out of time. We generate a fair bit of SAR. We synthesize over 1,000 analogs. Not going to point out the specifics there, but the two compounds I'm going to talk about, 11023 and 171, have the properties we want in terms of their non-agonist but ability to block serine 273 phosphorylation, and they actually have reasonable pharmacokinetics from an oral dose. This is the data here for 171. This is its binding. It's potent, more potent than Rosie. It's inactive in reporter assays, but it has this issue that it has slight PPAR alpha activity. It's micromolar on the human receptor, but it's in the 3-digit nanomolar on the mouse receptor, which might complicate the interpretation of the mouse data, and this is the ability to block phosphorylation. This is 023, which is a gamma-weighted antagonist that has no minimal alpha activity. In fact, this is essentially dead. Nice potency, again, non-agonism. So here's the data. We did a 21-day study in DIO mice, oral dosing once a day, 20 mg per kg for 023 and 3 mg per kg for 171. Here's the glucose tolerance test. We see good correction with 171 at this low dose, a better correction with 023 at the 20 mg per kg, as well as correcting fasting glucose. This is the AUC differences plotted here. They're statistically significant. Not quite as good as Rosie when you look at it from the AUC point, but in the fasting insulin, we're actually outperforming Rosie here at all doses that we tested with the compound. So we're real excited about this. More interesting, and this is emerging as an area of interest in metabolic disorders, we see a induction to the M2 characteristic for macrophage away from M1, where M1's being the pro-inflammatory and the M2's being anti. So with ROSI and R2 compounds, we see an increase in the population of macrophages that have M2 characteristics. If we just measure the inflammatory cytokines in the white adipose tissue, we actually, again, see a reduction. So we believe we're having the similar anti-inflammatory effect with these compounds. If we go to slightly higher dose with 171, here's the real home run in that in fasting insulin, we're better than ROSI and DIOs, and we even corrected in lean controls. If we then look at GTT, again, we have a significant difference in improvement over rosiglitazone in this experiment. More importantly, to distinguish ourselves as a novel mechanism, we see a different expression profile in white adipose tissue. We see no hint of agonism where ROSI would increase AP2. We see, if anything, a slight reduction with 171, as well as LPL. In the CDK5, the serine 273P phospho gene set, we see an improvement in adipsin. And consistently, we're not seeing anything with adipo-Q, but one could argue that this is a marker of adipogenesis, whereas this is more of a marker of insulin sensitization. You can't read this, but the NMR data suggests that we have no impact on the lean mass, and where there was slight weight loss observed for this compound at 20 mg per kg, it was actually picked up as a decrease in the fat concentration of these mice. However, there was about a 2.7 gram loss of fat. There was no impact on food intake. With ROSI, you always see an increase. You see an increase in body weight. So to rule out any type of toxicity, we dosed these compounds at multiple doses in lean mice. All those animals gained weight. There was no impact on feeding, and again, with both these compounds. So to summarize these guys, 171 is a dual weak PPAR alpha agonist that has gamma antagonist activity. So with the alpha agonism, we're seeing FGF21 increase. I didn't show that data, but we believe it should hold an advantage over alglitazar, which is a full agonist on gamma, which is known to be problematic, and it's a full agonist on alpha, which has been difficult in the companies that have tried to develop second generation full agonists of PPAR alpha. So we think we all hold an advantage, but we have a backup in that we have a selective PPAR gamma antagonist with no alpha activity. There's no effect on feeding, and we see very robust correction in fasting and insulin and glucose. So that was the PPAR story. I'm going to run through just a very few slides here, 8 slides maybe max. So if you can hang in there with me on our ROR gamma program. And initially the goal was to go after this as an anti-inflammatory molecule. So I tried to jam 4 slides into one as to what ROR gamma's role is in Th17 cell differentiation. It's the critical transcription factor for differentiation down the Th17 pathway. These cells produce IL-17, which have repressive effects both on Th1 cells, but also on Tregs. And they also have a role in peripheral clocks, other types of T cells, but clearly play a role in autoimmune disease and in rheumatoid arthritis, with the IL-17 driving additional cell types that are involved in tissue destruction. And just to point out that ROR gamma expression is increased in stimulated macrophage as well. So the way we got involved in this is we built a nuclear receptor library to help the chemical biology aspects around the receptors, hoping that we'd find ligands for receptors that had no known ligands. And when you do this, you have to validate each one of these constructs. So we had a small collection of known nuclear receptor modulators. This particular compound here happens to be an LXR alpha and beta agonist, and that's this data right here. It's robust. It had been published to also activate FXR and PXR, and we see that as well. But to our surprise, we saw this significant repression of RR alpha and RR gamma. So this got us started in this whole area where we wanted to take this compound and remove its LXR activity. That was pretty simple, removing this hydrophobic moiety from the sulfonamide and nitrogen. This forms a nice hydrophobic pocket in the LXR crystal structure, so that's gone. You remove all the LXR activity, so you have basically dual RR alpha, RR gamma inverse agonists. This is the compound we published in Nature showing efficacy in EAE. But then we wanted to get to RR gamma selective molecules to see, what's the difference in role in Th17 cell maturation if you have gamma selectivity versus alpha. So we synthesized or removed the sulfonamide to these biaryl compounds and came up with structures like 1555. This is our battery of acids again, assays again, just showing, the lab has to do all these studies as you progress compounds. And not going through the list, I just highlighted 3 compounds I'm going to talk about: 2211, 1555, and 9805. For the most part, in biochemical assays, they're all inactive on RR alpha. They're all fairly potent on RR gamma and devoid, for the most part, of any LXR activity. They have reasonable pharmacokinetics. So with that, prior to doing in vivo studies, we wanted to show that would they do what we would expect. So in EL4 cells, could we block differentiation and expression of IL-17A, and we do, as well as IL-23 receptor. And the Th17 cell differentiation assay here, again, we see the same thing, and this is intracellular staining, and this is Luminex analysis. And you see a nice dose-dependent reduction in IL-17, as well as the same thing in the intracellular staining. So the compounds are doing what we would expect. But we had an interest in wondering what's going on in macrophage. I don't have the figure here, but we can show that LPS stimulates the expression of RR gamma, which was not published. However, Ron Evans and Nurser had shown a series of nuclear receptor expression patterns in various cell lines in response to LPS, and you do see gamma going up. So we treated LPS-stimulated RAW cells with these various compounds, and this is where we started to see a difference between where 2211 and 9805 are equipotent and have similar pharmacokinetics, which will come into play in the next slide, but they do very different things. They both can repress TNF-alpha, but they differentiate themselves on IL-1 beta and IL-6. So we went to the CAA model. So this is a collagen-induced arthritic model. First in the prevention mode, where you immunize the mice and you start dosing drug. Dexamethasone is a flat line, as you would expect, as the gold standard. And in vehicle, you get an increase in inflammation in the paw. And this is what the paws would look like here. So these are scores that are made by the technician based on the number of inflamed joints. 2211 showed beautiful efficacy in the prevention mode. Now, the prevention mode, while this is nice, you're not going to take a drug hoping in anticipation that you're going to get arthritis, right? So you do the therapeutic mode, which is you immunize, and then you come in, and just before you boost the animals, you start your dosing. And what you see here for 2211, this is the vehicle, 2211, initially there's modest efficacy, but then there's this turn where we start to see a dramatic change in the efficacy of this compound. And this has been reproduced multiple times. There is a dose response at 8 mg per kg. There's no effect. So this is a compound effect. And the interesting thing to us, 9805, by all accounts, looked just like 2211, similar pharmacokinetics. It just had this differentiation in macrophage, and this compound essentially doesn't work in the same paradigm. So the question is why? So again, we turn to HDX. I don't have the answer from this, but if we look at 2211 versus 9805, they have very different characteristics in which the HDX pattern, particularly in this AF2 region of the molecule, whereas 9805 has no stabilization, whereas 2211 has. And not shown in, I don't have a slide for this, we now show that they impact their interaction with NCOR very differently, actually in opposite directions. So there's two different ways they inverse agonize this receptor, and we're following up on this now through mutagenesis. So brings me to the last few slides, and I'll leave you with interesting data, which I have no idea why it's working. So in those CIA models, you use DBA mice, they're susceptible to CIA. And what we did is, in addition to 9805 and 2211, we used this compound 1555, which was less potent than 2211, but structurally similar. Did the study, animals were opened up and organs taken, and the postdoc noticed that the 1555 treated animals actually had no white fat detectable whatsoever. Now, these are lean mice, so they don't have a lot, but it was clear that it was gone. So we decided to take this compound and put it in DIOs, and what you see is a nice decrease in body weight. You get a dose-dependent effect here as well. This is the animals open up. You can see the amount of fat is dramatically different between the vehicles and the drug-treated. If you let this go out to 45 days, this flattens and doesn't go below the controls. The interesting thing is there was a reduction in food intake, but not enough to account for this weight loss. So we went through and did additional characterization of these mice. We correct total cholesterol to that of lean controls, triglycerides, HDL. Again, in mice, cholesterol is almost all HDL, so if you reduce cholesterol, you're not going to see an increase in HDL. So what's going on? So if we took the inguinal fat pads and stained for UCP1 induction, we actually see it not throughout every cell, but it's a sporadic distribution, which would be consistent with the beige cell population in the inguinal pad. This is a 10x, just showing you that there are nice clusters. If we look at the bat, we see robust induction of UCP1 and PRDM16, which are key thermogenic genes. And then in the bat, we also see an increase in FGF21, whereas in the white adipose, we do not. So this is just a picture showing it. It's hard to see here, but if you look at the subauxiliary fat, it's essentially non-existent here in these mice, whereas the vehicles, it's abundant. You can tell by the bellies that they're different. The NMR shows that there's no impact on, well, the lean mass has gone down because the body weights have gone down a little bit, but the fat difference is dramatic. We've improved the fasting insulin dramatically. And then I'd leave you with that. We have the wonder drug here, but then you get the bummer data. So we do a glucose tolerance test, and we've actually made it worse with this situation. So the leaner animals at this point in time are not responding or not disposing of glucose efficiently when administered. If we take them off a drug, 10 days later, you get what you would expect. So they're lean, so you get a lean profile, and the animals that are still fat have the fat profile. So what's going on here, we're not 100% sure. We did look at quadriceps. We actually see a statistical significant increase in the grams of muscle in these mice. Again, consistent that we're not just wasting these animals. And the livers are dramatically reduced because of the steatosis here has been corrected. So we're trying to figure out the mechanism. So we go to our 3T3L1 assay. And what you would expect from a ROR gamma knockout, and this has been published, is that you would get an increase in AP2, so you get an increase in adipogenesis, but the adipocytes are smaller. While they're more in number, they're smaller and they're more insulin sensitive. They have improved expression of adiponectin. What's not from the ROR gamma knockout is an increase in FGF21, but our compound is a repressor of gamma, we see that. What we didn't expect is this increase in RR alpha. So there is an RRE in the FGF21 promoter, so maybe we're a partial agonist of alpha, increasing FGF21. We show that we're not binding to PPAR because anytime you see AP2, that would be your first hint. We show that that's not the problem. So again, scratching our head as to what's going on. Now, I mentioned that we had run these other compounds that are very similar to 1555. They're all aryl at this position. And so I had asked the DMPK group to go back and look at plasma. They found that there was about 10% of the desacyl metabolite, which we call 1590. They saw it first in hepatic microsomes, and then they found it in the mouse plasma. This is the data here. So this is the compound dosed. This is the exposure you get for of the metabolite. And then if you just dose the metabolite itself, you can get reasonable exposure. So you do what you would normally do. You scale this up, you put it in mice, and lo and behold, the mice are losing weight with the metabolite. Now, while this is great, in this case too, here we're seeing an increase in food intake for the animals on the metabolite. While this is real interesting and exciting, we now have no idea what the target is of this, because in ROR gamma reporter assays that's dead, we have to now go in and see if in fact it is a partial agonist of ROR alpha. So interesting phenomenon, doing a phenotype screen, postdoc making an observation, leading us into a whole new area for an ROR gamma modulator, which we had no anticipation of initially. So I'll leave you with that, where we have no idea. So if there's suggestions on how to find a target of a compound that looks real interesting, I'll be happy to talk to you. So just to summarize, 171 and 023 represent functional SPPARMs that have potential to be safety assessment candidates. These are under license by Ember Therapeutics. We need additional data to support the IND filing. 171 should be superior to alglitazar, and we're expecting the outcome of their results here. Everyone and their grandmother has stayed away from PPAR, but if this gets approval, we'll open the door wide open and opportunities for a compound that has advantages. 1555 or its metabolite demonstrated interesting anti-obesity effects in DIO mice. Perhaps 1555 is a partial agonist of ROR alpha, but now we're obviously looking more at the active metabolite. So the question here is, what is its target? So these are the people that did most of the work. Ted has done all the chemistry. Alex, this was at a party at my house, that was the best picture I could get. He's done all the animal work on PPAR. Mira Chang has done all the animal work on RORs and expression profiling. David Marciano, a graduate student in the Kellogg program, torturing my dog at the party. He's done all the PPAR, HDX, structure, function, and crystallography. Rest of the group, help from the Spiegelman lab and the funding to support this. And with that, I'll be happy to take any questions. Nice.